I now have the great pleasure to introduce this year's Nobel laureate in physiology or medicine, Dr. Gary Ruvkun, our second star. Gary Ruvkun was born in Berkeley, California. In 1973, he received a Bachelor of Arts with a major in biophysics from the University of California, Berkeley. In 1982, he received his PhD from Harvard University after studying bacterial nitrogen fixation genes in the laboratory of Frederick Ausubel. Dr. Ruvkun then started his postdoctoral research jointly between the laboratories of Walter Gilbert of Harvard University and Robert Horvitz at the Massachusetts Institute of Technology, remaining there until 1985. He then became an assistant professor at Harvard University and started his own laboratory at Massachusetts General Hospital. Gary Ruvkun is now at Massachusetts General Hospital and professor of genetics at Harvard Medical School in Boston, Massachusetts, USA. Dr. Ruvkun, we are very much looking forward to your Nobel lecture entitled A Vast and Ancient Hidden World of microRNAs Across the Eukaryotes. Please join me in welcoming Dr. Ruvkun on to the stage.
Thank you. Thank you very much. Am I on? Yes, I thought I might have unplugged. It's really wonderful to get this kind of reception. It's been a wonderful week, and the phone call from Stockholm was lovely. Thank you. So I'm going to talk about, our work on the vast hidden world of microRNAs, and it's many of my students and postdocs over many years. And I just want to remind you for a second that what we're talking about is watching these lineages. In wild type, they make a transition, and in a Lin14 dominant mutant that I'm going to be describing, or the Lin4 microRNA mutant, you suddenly fail to make this switch, and you keep going L1, 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 L1. Whereas if you don't have Lin14 protein, you never get to L1 and you go straight to L2. And this was the sort of the worm's blessing and curse, was to use lineages as a way to assess what a cell is wanting to be and trying to be. So these patterns of division tells you what the cell thinks it is in terms of time or space. But as developmental biologists, talking about lineage was very strange to the developmental biology community. And so we would go and present these sorts of lineage mutations, and at developmental biology meetings, all the other people who were working on cell-cell interactions would think we're very much interested in ancestry and who your parents are, and developmental biology was all who your neighbors are. And so worms were sort of an outlier in biology, but we kind of liked it. And that's that the worm community was always a little bit separate. And so the microRNA story is a bit of that, where we sort of think different, right? And it works to think different sometimes. And so the gain-of-function mutant was really interesting to study, and using the same kind of chromosome walking that Victor described, we got to the Lin14 gene. And the interesting part of it was not the protein coding segment, which is, again, a little bit strange. Usually what people are interested in is the protein. It's homologous to something that's related to a disease. And, and proteins are sort of have a lot more letters to them, right? There's 20 amino acids, and if it matches, it's significant, and DNA is harder to match. And these gain-of-function mutations turned out to be in the 3' untranslated region. So this is downstream. It's a 3.5 kb message. and about half of it is 3' untranslated. And there were 2 different activating mutations, and so that gave us a statistics of 2, which is way better than 1. And one of them was a 600 base deletion that activates the gene, and the other was a translocation that removes this back end of the gene. And if we look at the protein product, so we made an antibody, and this is the work of Bruce Whiteman and Ilho Ha, making an antibody to Lin14. It turned out to be a nuclear protein, and at the larval stage 1, it was abundant, and at larval stage 2, it was unabundant. And so it sort of fit with what the genetics was telling us, that the gene is active at the L1 stage and inactive at the L2 stage. And then the gain-of-function mutant failed to turn itself off, and it continued to have this nuclear protein at the L2 stage. And this nuclear protein didn't have any homology until a year or 2 ago, when AlphaFold was used to say that Lin14 folds in the same way as a couple of different things that don't have amino acid homology, but AlphaFold has saved our butts on having some homology, so we're going to the lecture tomorrow. Thank you, AlphaFold. And we can see this at a Western blot. Now, since Victor mentioned Southerns, it's time for an educational moment. A Southern blot is named after Ed Southern. Do you think a Western blot is named after Fred Western? No, it was a joke, right? A stupid molecular biology joke. And Northern blots aren't named after aren't we funny? No, I know. There's a lot of room to be better than that. So here's the Western blot showing the protein going down by a factor of 50 from L1 stage to L2 stage, but in a Lin4 microRNA mutant, we didn't know it was a microRNA when we started, it doesn't go down nearly as much, so it fails in that downregulation. And again, this is Bruce and Ilho did this experiment. Now, to prove that it was a 3' UTR, this is in the days before GFP, right? 1993, GFP is discovered, 97? You guys probably know. But there was no GFP, but LAC-Z fusions were the coolest thing then. So you could take beta-galactosidase from E. coli and put it in front of the Lin14 3' UTR. So this is an animal that carries a collagen promoter, LAC-Z, the Lin14 UTR. It gets turned off. Oh, I'm sorry, that should say L4. Oops. It gets turned off at a late stage. But in a Lin4 mutant, it fails to get turned off. Or if we switch the UTR to have a non-temporally regulated muscle 3' UTR, it fails to get turned off. So the Lin14 UTR has something that turns it off and is responsive to the Lin4 mutant. And this is the paper that was back-to-back with the Lin4 microRNA paper. And so here's the Lin14 stem loop. The Lin4 stem loop, and this is the microRNA itself. And that Victor's lab showed is upregulated at the L2. And the way that upregulation works is there's these 7 sites. You can see where they are relative to the Lin14 gain-of-function mutants. So this one is a weaker gain-of-function than this gain-of-function that gets rid of all of them. Here's site 1 that has Lin4 on the bottom, Lin14 on the top, and it's Watson-Crick base pairing with this loop, 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 and that keeps it from getting cleaved. So here's the model that emerges, that Lin4 microRNA gets upregulated and that downregulates the Lin14 protein, the translation of the Lin14 protein. And this, again, it was done by Bruce. Here he is today. and Ilho. So that's where we were in the pathway, but then LET7 emerged as having heterochromic defects as well, and so now I'm going to tell you the story of LET7 and Lin41 that happened a few years later. And so the LET7 phenotype is affecting a transition that takes place later, and what we're watching is the pattern of divisions of these cells along the lateral hypodermis of the animal, and these are cells that have different temporal patterns, and the last pattern is to not divide and to form these sort of racing stripes on the animal. And the wild type has a perfect racing stripe, and the LET7 mutant fails to develop those racing stripes because it keeps going L4, L4, L4. It never gets to adulthood, which some of us can understand very well. And so getting to the LET7 gene was the work of Brenda and Frank, and this was in collaboration with Bob Horvitz and Michael Basson, who did much of the genetics on LET7. And so they had to walk into the region and figure out that the gene corresponded to a microRNA, and we actually didn't expect it to be a microRNA, even though there was another microRNA in the Lin4 had already been discovered 5 years earlier. But we were imagining it was going to be the regulatory cascade, another set of transcription factors or something. But it turned out that it was a point mutation, and it was the point mutation helped us guide us because it said, well, that's the active site of whatever it is we're working on. And so when they did a northern blot, again, funny joke, northern, not southern, this microRNA is 22 long. It's not expressed at early stages, and then suddenly at the L4 stage, when we know it functions from genetics, it gets turned up. This is a lethal mutant, and so you can then do a genetic screen where now how do you work with a lethal mutant? Well, it's temperature sensitive lethal. So it's lethal at 25 degrees, but if you grow it at 15 degrees, which is fine for a worm, you can grow it up, do a mutagenesis, wait 2 generations, which is the glorious thing about a worm is you're talking a week. It's not 6 months to get 2 generations, it's a week. Or humans, it would be, 100 years. A week is better than 100 years. So you could ask for suppressors out, and out of that, sorry, where am I? Here we go. This is the work of Brenda and Frank, and they're in the audience too. And out of that, the suppressor turned out to be the target gene Lin41. So Lin41 is a conserved protein. It has lots of paralogs, both in the worm. These are all the worm genes, but these are the mammalian homologs, and it's a ring finger protein, probably a translational control factor. And when we looked in the 3' UTR of Lin41, we saw conserved complementary. So here's the Lin41 sequence going that way. This is the LET7. This is one LET7 microRNA, second LET7 microRNA. Similarly bulgy, not the same kind of bulge. This is more of sort of a 2-base bulge instead of a 5-base bulge in the case of Lin4. We don't know why that is. And so we have now a case of a microRNA here, the LET7 here, and in fact, there's paralogs of LET7 that I won't talk about that do these transitions here. And you can see in the lineage of Sulston that there's these waves of division. So here, there's one wave of generation of cells here, a second wave of generations here, a third wave, a fourth wave. And those are all the in-between molts. So at this point where there's no divisions is when the animal sheds its cuticle. And this idea of having molting is not unique to C. elegans. There's a whole large clade of animals called the ecdysozoans because they do what's called ecdysis, which is to shed your cuticle. And that's how insects, you go from larval stage to the larval stage as well. So there's something about microRNAs and larval staging for sure that comes out of C. elegans. And so then we could look with the LET7 sequence and look for homologs. But here we're aided by the emerging genome sequences in 2001. And so at that moment, there's a human genome that's available. And in fact, our hero, John Sulston, who did all this lineage work, so he did this sort of working almost alone, maybe 2 or 3 people, including Bob Horvitz, working on this, but it wasn't a giant team. And then he aids us, as Victor said, in sort of chromosome walking. And I have to tell you that I was important in this because Sulston came and saw me doing chromosome walking and said, "What a waste of a brain." That you're a pretty smart guy and it's so boring. I could walk from 100 places. You're walking from one place, I'll do it from 100. And so that's why Victor said we would communicate with them and they'll help us, and he did it with everybody, putting together the Genome Project as a gift to the field. And there's such a thing as thinking with your hands, and so he realized how genomics worked with he and Alan Coulson, basically doing cosmet preps and fingerprinting, a very, labor-intensive stuff. He's not sitting in a room thinking. He's thinking with his hands. And so at that point, he ramps up to do the Genome Project, and he goes from 2 or 3 people to 100 people. And in fact, he's a third of the human genome, doing a third of the human genome. And so when we wanted to look for a homolog, we could just take this very short DNA sequence and do it was before BLAST, but you would just say, well, is there anything that looks like this in the third of the human genome? And that was done with a modem from home. So I'm sitting at my desk and you get the data and it comes back and it says, yeah, there's a perfect homolog in C. briggsii, of course. Well, that's not a big reach, but zebrafish, human. At this point, all there was human, right? And zebrafish and all these other chimps, et cetera, they show up in the next 5 years, right? And now there's 20,000 eukaryotic genomes. There's another 500,000 bacterial genomes. I can't begin to, you all maybe know how wonderful it is to be a molecular biologist today, right? That you have all this data that you can just look at and like, oh, I think I'll blast this. It's all I do. So there's Amy, and of course, guess what that is? That's the LET7 sequence. And she's now and she lives in California, so you can, that's how people are in California. I'm from California, I know. And here's Brenda again as working on this project as well. And both of them are here today. And then how about proving it, right? So that was a database search, modeming into a third of the human genome. So part of this project was me being charming and begging for RNAs from people. And so Mitzi, who's also in the audience, sent us fly RNA, staged fly RNA, and Amy did northern blots. I won't belabor the joke. And you can see that there's nothing early, and then it comes on at sort of the equivalent of the L4 stage of a worm, if you don't know too much biology. And so you can see that this is a late stage regulatory event. It doesn't prove that this does anything. It just says that it's regulated temporally in the same way. And in the Lin4 case, it was not, you don't find a homolog in the fly. And we could also do it in zebrafish with RNA that Mark Fishman sent. And here's LET7, not there in the early fish, but sort of later in the development of a vertebrate, right? So that's getting closer to a human. It's still kind of it's a fish. but it's temporally regulated, and it sort of validates what the human genome comparison said. It's just, but it adds to it that it's temporally regulated. And then we can look in human tissues, and you can see that it's in the brain or kidney, liver, et cetera, and sort of less in cells that are more totipotent or dividing and not so differentiated. But it for surely, what it did say is that it was expressed as a 22 nucleotide RNA as well. So summarizing here all the organisms we looked at, and again, people sent us, RNAs from just the weirdest organisms. Super fun. Some of them came from Australia. So plus means we could see a band. We didn't get developmental series from everybody, but almost every place we looked that's in the bilaterians, and these are the ecdysozoans, but almost all of them have it, and almost all of them are developmentally regulated. And we did not detect LET7 in these more basal eukaryotes, but other microRNAs have been detected in these, and so LET7 didn't happen to be conserved. So this told us that microRNAs were going to be in many different organisms of the eukaryotes. And then in the end, we have sort of hundreds in the vertebrates, almost a couple hundred in the invertebrates, a few in sponges. There are no microRNAs in the yeast, but plants have also hundreds, and none of the plant microRNAs are equivalent to the animal microRNAs. So what it tells you is that the common ancestor of animals and plants had microRNAs, and I'll tell you about argonaut in a minute. They use the same peewee protein, the argonaut, to present those microRNAs, but they've evolved different microRNAs, and it's either because evolution is too fast, but it's really only a factor of 2 between plants and animals. And so it might tell you that there's something different, and what I think, and I'll probably get to it, is that the time scales for how it works in a plant versus an animal is very different. We have microsecond things going on in our nervous systems, and maybe I'm just an animal-centric view of the universe. Maybe plants don't have millisecond type things going on. Their sort of temporal sequence of responses is slightly more slow, and that might be what's different, and I'll get to that in a bit. So what are some other conserved microRNAs? It's not just LET7. Another really highly conserved one is called miR1, most likely because it was the first one. I don't remember who discovered miR1. Probably Victor. Oh, there it is, yes. Sorry, Victor. It's hard to keep track. So this is really highly conserved, and I bet it wasn't the first one you discovered. You just put it top of your list. And actually Victor's lab shows that it's involved in muscle development, and it does something in our heart muscle in a mouse. And we did some work on it as well. One of the ways to study these microRNAs is to take the gene for it. And even though this is coding for a tiny RNA that folds back, et cetera, you can still slap GFP onto it. We had evolved by 2001 from LAC-Z to GFP because Marty Chalfie had made GFP what everybody uses. And you can see now that this is expressed in body wall muscle and the muscle of the pharynx of the worm. So it's sort of a muscle-specific microRNA. And in fact, if you look at who the targets might be, you can see good conserved base pairing, that the miR1 is conserved, as is the targeting of a acetylcholine receptor called UNC29. UNC is used to refer to worms that don't move. They act uncoordinated. And in this case, the uncoordinated is due to a muscle defect, a response to a motor neurotransmitter. And these cholinergic acetylcholine receptors are multiple transmembrane domains with multiple domains, multiple protein domains that heterodimerize. And in fact, in collaboration with Josh Kaplan's lab, we can see that the UNC29 protein is expressed at a lower level in wild type, but when you get rid of this microRNA, it now is expressed at a much higher level. And what was strange about this is that we then expected the microRNA to be, microRNA mutant to make more of that protein, to therefore be more resistant to levamisole, and we got the opposite result out of that. We don't know why that is. One view is that there's other acetylcholine receptors that are also repressed, and there might be a relative mixture of the two that leads to the levamisole sensitivity. We also looked for more microRNAs. John Kim did this in 2004 in collaboration with Ken Kosik's lab with Anna Kraszewski, pulling out microRNAs from cortex. And we got miR-124 as one of the most abundant things that came out of that. And there again, we could fuse in GFP. This is very highly conserved between mouse and worms. And with a GFP, we could see that it's also neuronal specific. And in fact, miR-124 in a mouse is in the cortex and cerebellum, very particular to the brain. And when we looked at where is that microRNA on a polysome prep, so this is where monosomes are here, polysomes are where translation is really cranking out the product because multiple ribosomes have glommed onto the RNA and are gliding along, producing protein, multiple copies of a protein from a single mRNA. We can see that the microRNA is actually in the polysome fraction on a sucrose gradient, saying that maybe it inhibits translational elongation and not translational initiation, which is something I'm really interested in today. So I wanted to do 2 minutes on plant micros because they've been really interesting. So at the same time that the Bartel lab and the Ambros lab and the Tuschl lab were cloning microRNAs, this is a Bartel paper, and notice Brenda Reinhardt's on this one too. So Brenda's kind of the star of the show here. She was involved in this plant micro work, and one of them, called miR-156 from a plant, is very highly conserved in plants, all the way from Arabidopsis to rice. And rice is a long way from Arabidopsis. It's monocot versus dicot. These are like as far apart as you can be on plant phylogeny, and it's still conserved. And if you look at where it is, some of the complementary sites to the microRNA are in the 3' UTR. That's the light box here. But a bunch of them are actually in the protein coding region of the plant. And it's all perfectly conserved, and the base pairing is perfect base pairing. It's not the base pairing that we see with the loopy, bulgy base pairing in animals. And that's a blessing in terms of finding targets, because 4 to the 22nd power is a lot more statistically significant than 4 to the 15th power. And so it's easy to find targets. And if you look at who the targets are, there are a transcription factor called SBP, and this is the coding region of it, and that's what it codes for. And so you can see that the same thing is coded. Here are the ones that are in the 3' UTR. It's again, probably that's driven by a selection to target this coding region. And what's interesting about the plant micros is that they tend to work in a much more RNAi-like fashion of leading to degradation. So again, at the time that this micro explosion was happening, was the explosion of RNAi. And sort of a key event was Craig Mellow's discovery that there's a protein that it's called a Pee-wee protein that mediates the presentation of these 22 nucleotide siRNAs in the RNAi pathway. And so the question was, do microRNAs use anything similar? And it's almost numerological reasoning, right? Because we're saying, well, this is 22 long, and this is 22 long, and don't you think they're the same? And, in my 22 is the number of letters in the Hebrew language, and so, I used to bring people in and say, well, it's a magic number, and there's a whole ideology around 22 in sort of ancient Judaism. The people in my lab were like, is he crazy? And the answer is yes. And so here's Dicer. It's sort of course it should interact with RNA. It's got an RNase 3 domain, and those trim off from the 3' end of a message. And so that's involved in production of siRNAs. And then this is what Craig discovered, RDE1, and then it turned out that RDE1, so this is a program that we developed in my lab, I'll tell you about it in a second, where we take the protein domains of RDE1 and we do a blast P against everything in the animal, many things in the animal kingdom, many things in yeast, many plants, many protists, sort of 500 across. So blue means there's a homologue, and a good one. This isn't weak homology. Bright blue is like, yeah, you'd believe it if you looked at it. And so you can see that the RDE1, Pee-wee, is found all across the animals. There's a couple of weird guys that don't have it. And then what was really important in the history of our work is that these proteins, the Pee-wee proteins, of which then these are all the worm paralogs. So these are different Pee-wee proteins. These are the ones Victor mentioned, ALG1, ALG2. These are the ones that mediate how microRNAs work. These other ones mediate other RNAi pathways. And the worms are sort of lunatics about Pee-wee proteins. They have 24 of them. We humans have 8. Plants have 12, 15 sometimes. Worms are really crazy on them. But what was really important here is that Saccharomyces cerevisiae doesn't have this, and that saved our asses, because if the yeast geneticists had been able to work on RNAi, they would have eaten our lunch. They're just so damn good. And so thank God. But it's also interesting, why did these clades of yeast choose to delete it? And I won't get into it, but they dumped 99% of their introns. So yeast they have 100 introns left, and we have 100,000 introns. And so it's part of their trimming down of their genome. Whereas Saccharomyces pombe, Schizosaccharomyces pombe, has argonaute proteins and does RNAi. And it's not a coincidence that RNAi was discovered almost coincidentally in plants that have the argonaute proteins and fungi that have it, as well as in nematodes. But even more so, and this is the program, if you want to use it, you can just type into Google DepCod, and this was developed by Yuval in my lab and Ruslan, who's a collaborator at Mass General Hospital. So now we're going to go into using feeding RNAi, and the discovery of RNAi by Mello and Fire was super important, but the discovery of feeding RNAi made it easy for all the rest of us to do it. Instead of having to inject double-stranded RNA, Julie Ehringer's group at the Medical Research Council figured out, and it was Lisa Timmons in Andy Fire's group who actually figured this out, that you could put in any worm gene between a T7 RNA polymerase promoter, so you make double-stranded RNA in a bacteria. and induced by IPTG. And you have to be really old to know what IPTG is, but some of you know. And that will induce this double-stranded RNA in the E. coli. The worm eats it, and the double-stranded RNA goes from the gut to the rest of the animal, and RNAi works. If you propose this in a grant, he'd kick your ass off no way. But it works really well. And so they made these libraries and sent it to us in around 2004. And so we could then just feed, and I'll show you what it looks like in a bit. And so we did feeding RNAi, and this was first with Craig Mello's lab, where we could take dicer and do RNAi on dicer, and it looks just like a LET7. Or we could take ALG1 or ALG2 and do RNAi, it looks just like a LET7. That's not enough to say it's the same, but we could also look at what happens to the Lin4 microRNA. So this is what it looks like in wild type if you're not feeding it anything. But if you get rid of dicer, now you have less of the mature and you have the precursor accumulating. And the same thing with Argonaute-1 and all the other peewee proteins here that don't have anything to do with microRNAs. So just these two have something to do with microRNAs. And it worked on a second microRNA. It's not just Lin4, we could use it on any of them and they would accumulate a precursor. So one last thing to talk about RNAi is that there's a weirdism of RNAi in some organisms. So the RNAi that was studied in Drosophila or in humans, like us, is an RNAi that uses dicer and produces siRNAs that are the same length as the microRNA. But then there's a secondary amplification process called RNA-dependent RNA polymerase. And that takes a primary siRNA that's produced by this pathway, uses it as a primer in the same way that a viral replication works by priming, in the case of a virus, from a tRNA. But this is an RDRP that makes copies, and you end up with 1,000-fold more siRNAs if you have an RDRP. And worms turn out to have 4 of those, and one of them came out of a screen for enhanced RNAi that I'll tell you more about. And the other ones are important for sort of making RNAi work, so that if you inactivate this one with a mutation, we didn't do this, it was Ron Plasturk's lab that did it. They showed that this was important for somatic RNAi and the other for germline RNAi. So this is looking at the pattern of who has it and who doesn't. So this is taking the RDRPs, and what DEPCOD does is it says, okay, I got a pattern of blue and white. Who else has that pattern of blue and white? And so all the RDRPs look the same. And then the ammonium transporters are sort of cousins, and maybe they act in the same pathway. And phosphoglycerate mutase looks similar, and maybe that acts in the same pathway. I'm not going to go over that. What I'm going to go over here is blue is who has the RDRP. So this blue, bright blue, is the worm finding itself. And all these other blues are animals that have RDRPs. I've told you that plants have them, and they use them to extend from the microRNA. But a whole bunch of animals like us don't have the RDRPs. And it's, again, not a coincidence that RNAi was discovered in the organisms that have this 2-stage. So it was discovered in plants and fungi and animals. But what are the animals that have it? So, this is an arachnid. This is a spider. I'll show you a list. So again, this is our DEPCOD way of looking at it, but you can also just do classic BLAST-P, and most people don't know, but when you do BLAST-P, if you go to the taxonomy tab, it will tell you who has it in order of how homologous it is. So the top hit, and here I'm excluding worms, so the top hit are these sort of mud-dwelling protochordates, so that they have the best homolog. And then we get down to scorpions and spiders and mites, and it's like you're 13 years old and it's just what you want to read about is spiders and ticks and mites, and they're badass creatures. And so what it means is that our worms are a lot more badass than we realized, right? And that we should respect their badassedness. And of the animals, it's again, horseshoe crabs, and what do they have in common? They tend to go hypoxic a lot. So like the bivalves, when the tide goes out, they close up, and they're hypoxic during that time. And plants have this, and plants spend a lot of time hypoxic because they get flooded when you water them, right? And I have no idea why that has anything to do with antiviral defense, but I'll bet it does, maybe, or not. Okay, so I'm going to end with about 10 minutes on, and how am I doing for time? Oh yeah, I got 6. Okay, that's approximately the same. This is a screen that Sylvia Fisher did. Now, I've told you that worms are really good at RNAi. We can do feeding RNAi. What a gift. But we still thought we could do screens to make RNAi better. Can we get mutants that have RNAi that's even more than normal? Which is kind of loony because it's so good. But another view of this is that RNAi is an antiviral defense, and maybe animals sort of induce or don't induce their antiviral defense depending on whether they feel like they're under a viral attack. And so maybe if we go for mutants that are enhanced for RNAi, we're putting the animal in an artificial state that would normally be very transient. It will only happen if there's a viral infection. But now, because of a mutant, we're putting them in a state much more all the time. And so that's why it's sort of not stupid to go for things that are improved. The normal way you learn genetics is this is all the ways to break an organism. But really, the power of genetics is it's all the ways to switch things on that shouldn't be on, or switch things off that shouldn't be off. And that's what physiology is all about. It's all about, when do things get induced and not induced. And so these screening for enhanced RNAi is sort of trying to get at that, and I'll tell you why and how and why. So we start with a transgene that's a collagen, and it makes the animals roll on a plate. So they crawl around and they do these little circles, and if you don't do RNAi, this strain is rolling just fine. And if you have a mutant that makes RNAi work better, and this happened to be an ARE6-7 that was one of the hits, now the animals crawl away. And so you could just do a screen for things that aren't rolling. And so out of that screen, Sylvia did it, and these are the hits. And she gets sort of 70 hits out of this, and this is in order of who's best. And the ones I'm going to tell you about are Lin35, Lin9, Lin54, but there's others as well. But these are the ones that are sort of fun. And she's now at Harvard Medical School studying these too. So these are some of the hits, and for example, these are called the SynMov-Bs, and I'll tell you about these in a bit. So if you take a particular double-stranded RNA that doesn't work very well in wild type, it barely causes lethality. We don't know why, but we know that in these mutants that are in what are called the SynMov-Bs, now RNAi works way better, and we get the lethal phenotype almost 100%. And what are the SynMov-Bs? So these are mutations that were originally identified in the Horvitz lab. I'll tell you a little bit about them. And so a wild-type animal will make this vulval protrusion that's involved in mating with males to then fertilize the eggs. And that's normally made with one. And the way it makes one is by an anchor cell that's adjacent to this hypoderm, sending a signal. It's a growth factor signal. And so they were interested in this because this is a RAS signaling pathway. But the mutants that they got And these mutants were presented by Chip Ferguson in Horvitz lab group meetings when Victor and I were graduate students, and we would hear all about SynMov-A's and SynMov-B's, because the original mutant that was isolated was a SynMov-A, SynMov-V double, and it caused this extra MOV. And then the Horvitz lab did amazing work over about 10 years, 10 different papers, showing that you need to have both of them knocked out. And what that leads to is too much of this growth factor from the hypoderm, not from the anchor cell. And it's kind of just weird stuff. But then we get all the SynMov-Bs are enhanced for RNAi. And what does that have to do with this? We'll get to that in a minute. And there's Bob, and this is our Victor and my mentor. And this SynMov work is sort of number 8 on his list of, what people will say major discoveries, but it's utterly awesome stuff, wonderful papers. And of course, Bob has been important in our early work, but it's coming back. This is all in the last 2 years, right? So what should I work on next, Bob? Please. So what do these turn out to be? So Lin35 is the gene that's sort of the most sexy, and that's retinoblastoma, and that's an oncogene that's involved in retinal tumors, one of the very early oncogenes. But there's human homologs of Lin9, Lin37, all these things have homologs, and what these do is they sort of assemble onto what's called a dream complex. And I had originally kind of resisted working on this because it's sort of chromatin regulation. I was really worried that I will never make sense of chromatin. Me, others can, but I wouldn't. But we've gotten more and more interested, and I'll show you why. And this was the experiment that really made it fun. So we started with a Lin8 mutation. So that's a SynMov-A. It has no phenotype on its own. And normally, in order to get it to show a MOV phenotype, you have to do something to knock down a SynMov-B. And we decided, well, gee, we know that if we knock down a SynMov-B, the animal is enhanced for RNAi. And so maybe if we do a virus infection, so the Orsay virus was identified by Marianne Felix from an orchard in France, and so you could infect a worm with Orsay and ask, if we take a Lin8 mutant and infect it with Orsay, do we get a MOV phenotype? And so if we take that Lin8 mutant and don't infect it, nobody's MOV. But if we take that Lin8 mutant and infect it in 3 separate replicates, we basically get 10%, 10%, 10%. And so that says that a viral infection essentially downregulates SynMov-B somewhere. And this is what a SynMov-A, SynMov-V double, it's 100% MOV. And so the idea is during a viral infection, this SynMov is being turned down, and that helps the animal resist the viral infection because RNAi is enhanced, and that's what you want during a viral infection. You want to be resistant to a virus. And in fact, we could see it when we look at the amount of RNA by RT-PCR. So a wild type has sort of this much, 1, and a Lin35 mutant that's constitutively airy resists the infection much better. It's about 20-fold better. And a Lin15B, which is another SynMov-B, resisted about 100-fold. So it says that this is a natural state, and the reason they're airy is because we're constitutively putting the animal in an antiviral state. And antiviral states are important. It would have been better during COVID if humans were in an antiviral state. The animal loss of the RDRP seems like a bit of a mistake. Maybe it's because we have a nervous system, because it seems like plants didn't lose their RNA-dependent RNA polymerases. Okay, I'm going to skip this last bit. And I want to go to talking about the people in my lab. So this is the lab today at our house, and the work I talked about just now is from Ashwin and Himani. and there's other projects I didn't go over. And this is the list of people in the lab over the years. And I want to mention, of course, there's a lot of names. I won't go through all of them, but one of the things I want to mention is, you know, I've lots of times I go and I give a talk, and they say, talk to us about mentoring. And my lab has about 800 person years of people. So, you know, most of these people were in the lab for 6 years approximately. And so they, you know, multiply 6 times the number of people, and it's 800 person years. I'm 40 person years. And even if I say, oh, I'm really good, so maybe I'm worth 80 of those 800, that's still 10% of the lab. And when people leave my lab, there's a hole, right? We lose talent and knowledge, and the people who spent time in my lab remember each other a lot and are friends forever, and they write papers together 20 years later. You know, it's just the most wonderful thing to see. So the mentoring in a lab is the lab. And you know, yes, I'm an important part of it, but I'm not at all the most important part. And I want to mention Diane Sacchetti, who has been the soul of the lab for 26 years and has been really important in all the people. She cares a lot about all the people. And many of you might have seen the famous ice cream truck scene that's on Twitter, and I don't know, got 1 million hits of, you know, me the day of the Nobel announcement serving ice cream. It's because her husband, Frank, who's the family friends, has an ice cream truck. And of course, he came on October 7th with the announcement of the Nobel, and we served ice cream to everybody at Massachusetts General Hospital. Super fun. And this is the home. There's Natasha, my wife, at the Rolling Stones concert, at the famous Food and Books. She's been the key part of the lab, my life. And my lab has always had a theory that she's the brains of the operation, and it's true. I learn a lot from Natasha. Our daughter, Victoria, there she is in medical school, and it turns out there's an instinct to be excited when your kid's a doctor. It's really exciting. And Victoria's been the light of our life, and there she is getting her degree from Ambros, and Natasha was able to give her her degree because Natasha teaches at Ambros College, history of art. And there's Victoria with our dog Barnaby, who we talk to more than anyone in the house, in a high voice, a lot. I wanted to thank my parents. My dad went to the University of California, which is why I went to the University of California, as did almost all my cousins. And he was an engineer for many years, and that really taught me how to think about technology. And I sort of I didn't become an engineer, but I could have, should have. And my mother did not go directly to college. She came out of high school and went with her sister to the Young Hebrew Women's, like the equivalent of the YWCA in the old days. And that was where a virtuous girl at age 17 in her case, and she went to the San Francisco Young Hebrew Women's, whatever it was called, and met my father's sister, who met my father, and one thing led to another. And we did our homework together, my mother and I. That's me at a science fair project. I blame my parents for the terrible orbital mechanics of all the planets being in one orbit. This is inexcusable, but I was only 6. And this is my early work was becoming an electronics thing. That's what nerds did before the internet, before computer programming. And I learned electronics, and it really taught me a lot. And then just a quick sort of, yeah, this is going backwards in time, lab and home, important. Harvard and MIT, important. I spent a year on the road in South America. That taught me about surprises and gave me 100 stories to regale people with. And they might, it seems like after 6 years they get tired of them and leave. But it works for 5 or so. And then I lived in my van for a year at Berkeley. I did physics, and what that taught me were scientific revolutions. And not that I participated in them, but I got to read about them. And physics is taught historically, which is really important because it teaches you sort of the temporal sequence of how things are learned, and it sort of prepares you for revolutions. And when I got to Harvard in 1976, it was obvious that the recombinant DNA was a revolution. And now with genomics, that's the result of that revolution. And it has been a revolution for me, for sure, and many of us. And it really all started with space program. That was propaganda to tell the Soviet Union that we were going to win or send missiles towards them. But what it really did was inspire a whole bunch of nerds of my generation to become scientists. And that's why we have Silicon Valley in the US, and why we now have a good pharma industry and good biotech, and a whole lot of us were inspired by that. Lastly, as a kid, you sort of say, well, who am I like? And so I sort of bonded with all the people on TV. And so the early TV and radio was very Jewish. And as a Jewish kid, I said, oh, maybe that's like me. And so I wanted to be funny. And so I try. But I think funny is not just for comedy. I think funny is a way to look at the world at a slightly different angle, and that's very scientific. And so I actually think one should use that as a criteria for selecting postdocs. How funny are you? And I think that's it. Oh, sorry, Fred. Asabel I want to thank. That's how I started in molecular biology. That was my PhD advisor. He worked next door to me at Massachusetts General Hospital for many years. And Scott Kennedy, who was a postdoc, gave me a deer head that he shot. And Fred was in the office behind me, and I tried like hell for many years to find a deer butt to put in his office, because I thought it was appropriate for somebody who's your former boss. But you can't do it with like a junior faculty member. That's abusive. But I never did find a deer butt, but I always looked. Wally Gilbert was just this luminous figure in the Harvard biolabs that sort of a role model of how to be a scientist. And there were many people like him at Harvard that really inspired me, and over the years sort of taught many of us how to be scientists. And lastly, my department was funded by Hoechst Pharmaceutical Company because of the work of Hans Hermann Schörne and Ernst Winokur. I'm actually the Hans Hermann Schörne investigator at Mass General Hospital. And that was because Howard Goodman had cloned insulin. They produced insulin, and they thought maybe they could have that they could support it. And it was really because Dr. Schörner had a lot of pro-American feelings after World War II, where he had been a 15-year-old soldier in the army in Germany and was treated well by the US during the post-war period. And so I was a recipient of, World War II didn't end at the time that I was working, and it led to wonderful support in my department. So thank you very much.